In criminal investigation, the earth is frequently used as a hiding place. Preparators often buy weapons, garments, or fluid-stained objects to conceal evidence or deliberately accelerate biological decomposition. High-profile investigations, such as the buried body case in the United States, the 2000 Fortaleza case in Brazil, or the J Canal investigation in Spain, prove that subsoil concealment is a recurring hurdle in global forensics. The central problem in this case study we are analyzing is how the subsoil environment compromises forensic identification. Specifically, how does a burial process impact the degradation of genetic material compared to evidence left out on the upper surface? This case study evaluates a critical forensic experiment designed to establish a concrete timeline for DNA profile decay and filling a vital gap in current legal medicine.
To critically evaluate this phenomenon, researchers engineered a controlled outdoor experiment from January to March, approved by the Ethics Committee of the San Carlos Clinical Hospital. They utilized an active, nutrient-rich universal flower premium substrate to mimic a common urban domestic burial environment. Blood and saliva samples collected from two male and two female donors were deposited onto a UV irradiated white cotton t-shirt. To isolate variables, half of the sample were buried under 11 centimeters of soil, while the remaining half were exposed directly on the surface. For 10 consecutive weeks, environmental factors were measured every single morning. The surface environment maintained a dry profile, humidity A, with a peak temperature of 26.9 degrees Celsius, while the buried environment was consistently damp, humidity B to C, with an average temperature of 9.4 degrees Celsius and a stable, slightly acidic to neutral pH of 6 to 7. Prior research by Kowa and Bogas notes that these exact combined factors trigger rapid microbial proliferation, which directly targets the fragile cellular matrices of biological fluids.
The data revealed a severe non-linear degradation curve. In nuclear DNA quantification, the internal PCR control flagged indeterminate result as early as week 1 due to severe soil inhibition. By week 4, DNA quantification dropped to zero across almost all samples. However, subsequent SDR amplification using the identifier plus PCR kit bypassed this hurdle by diluting the extracts, allowing us to view the true macromolecular damage. The absolute genetic outcomes were striking. Surface blood samples showed the highest structural resilience, maintaining lower allelic loss during the first 2 weeks. Conversely, buried blood degraded immediately in week 1, losing an average of 27.75 out of 32 alleles. Saliva degraded rapidly regardless of location, primarily because it lacks a protective cellular matrix like blood. By week 5, an average total loss of genetic material was reached, and by week 6, 100% of the autosomal markers across all conditions were completely and irreversibly destroyed. Statistical analysis verified that biological sex played no role in this decay. When evaluating alternative laboratory methods for such highly degraded profiles, standard autosomal STR profiling clearly fails after 1 month. Viable alternative actions include utilizing mini STR analysis, testing advanced SNP sequencing panels, or pivoting to maternal mitochondrial DNA mtDNA tracking, which survives environmental stress much longer than nuclear material.
Based on this definitive outcome, Our group has formulated a critical three-part action plan for forensic investigator and crime scene technician handling buried material evidence, which is the first one is immediate extraction protocol. Because buried blood and saliva face an irreversible viral window that averages just 5 weeks. Since involving soil destruction must be treated with maximum temporal urgency, evidentiary items hidden in topsoil must be prioritized for collection ahead of surface elements. Second is modification of lab quantification steps. Since soil inhibitor like humic acid cause false negative during DNA quantification, laboratories should not abandon samples that read indeterminate. Instead, they must proactively incorporate bovine serum albumin, which is BSA additive, or execute strategic extract dilution to neutralize soil inhibitor prior to amplification. And lastly, the third one, environmental mapping, which is forensic unit must document local soil composition immediately upon evidence recovery. So, in conclusion, this study successfully established a vital baseline, which is soil environment can quickly level the decay rate based on different biological metrics. In the race against microbial degradation, time is the ultimate adversary, making immediate collection and tailored extraction protocol essential to unearthing judicial truth before the genetic evidence vanish forever. And that's all from us. Thank you for listening to our presentation. We hope everyone have a good day. See you next time.
I just had to let you know you're